My name is Dr. Derek Fisher. I came to Southern Illinois University in 2012, so I've been here going on 9 years, I guess almost coming up on 10. So I got my start at a small liberal arts college in central Pennsylvania, Lebanon Valley College, and the summer after my freshman year, I had the opportunity to partake in undergraduate research. From that summer on, I did research every summer as well as through the school year. It was a biochemistry-related project. I was a biochemistry major, and I fell in love with science. Getting to work hands-on with faculty, other students, to make discoveries was just amazing. And to think that that could be a profession was something that I really hadn't thought of. And from that moment forward, the decisions I made were to put me on that path. So I went on and did my PhD work at the University of Pittsburgh. It was at the University of Pittsburgh that I started my work with bacterial pathogenesis. So I worked on an organism, Clostridium perfringens, which makes a bunch of different toxins and is a potent pathogen of both humans as well as animals. So after my postdoc, I started here in 2012 and got my lab going. So in my lab, we focus on the important human pathogen Chlamydia trachomatis, which is the leading cause of reportable bacterial sexually transmitted infections. And we're particularly interested in how this organism is able to cause disease in the host. There are specific proteins that we're trying to figure out the role that they play, with the idea if we know the role they play in disease, we can figure out ways to disrupt those roles to identify new approaches for preventing or treating infections. So in my lab, we do a lot of cell culture work, we do protein expression work, we do a lot of molecular biology that involves undergraduate as well as graduate students. So we kind of have this nice working relationship amongst the students. Let's go now and take a look at some of the equipment that students would get to use in the lab and the type of experiments that they would do. So in order to see our bacteria and to make more of our bacteria, again, we have to grow them in cell culture. So we can do these 96 well plate type formats to do a variety of different growth assays. So these are the types of assays that undergrads and graduate students would be doing in the lab, where we infect cells and then we monitor growth over time, and we can vary conditions to see how different conditions affect growth of the bacterium. This is an individual cell. Within that cell you have the cell nucleus, and then this structure here that kind of looks like a lima bean, that's the inclusion, and all these green dots are chlamydia. So that's essentially what we're monitoring. So we can use these types of assays again to figure out how our bacteria are doing. Under different conditions, we can make bacterial mutants and figure out ways essentially to slow down growth, which allows us then to figure out ways to prevent pathogenesis, which you could think of in terms of preventing infection or treating infections. So the types of projects that we have in the lab for the genes or the proteins that we're interested in would have an undergraduate or a graduate student actually take that genetic information from chlamydia, put that into what we call an expression vector or a plasmid, and then they would take that piece of DNA or that plasmid and move it into E. coli and ask E. coli to make that protein. At the end of the day, we need to get that protein from E. coli, but E. coli is a much simpler system. So again, these are the types of things that undergrads and graduate students do in the lab. A beginning undergraduate student would have a small slice of this. They might initially start by helping with cloning or helping with the purification process. As they're in the lab more, then it grows and they get more steps along that pathway. And we also do cell culture in the lab. So graduate students would do the cell culture aspect, work with the protein purification, do protein assays to ask about the activity, so they have a much more holistic project. Starting undergrads get a piece of that, and then as they're in the lab for a while, that will grow. And I've had senior undergrads that would have a more integrated and a full project looking at various steps of chlamydial biology. My lab is always looking for enthusiastic graduate and undergraduate students that want a real-world research experience. If you want more information on what my lab does and how you can get into my lab to do research, please click on the link below.